We're good to go. All right, so welcome back.
Yes.
Everybody had a good break? Yes. So somebody brought the whole forest for us. No, it's good because I can actually use a couple of samples so for me to make it work. So what I'm going to ask as we start the experiments, we're going to do 2 experiments.
With the
first experiment, we're going to assess the number of stomata within our leaves. So we're essentially going to be looking at slides that look something like this, right? So the steps are actually not too difficult. You essentially have to take a little sample, put nail polish, let it dry, put some tape, clear the tape, and attach the tape to the slide. So I'm going to do this, and I'm going to ask our assistants to come to the front, watch me do it, and then the assistants are going to go back and do it with each table. And in the meantime, organize your leaves, figure out their species, start writing out a chart. So when you start looking at your samples, you can record the density of stomata for each particular species.
So
And we'll plan to spend about 1 hour per experiment. Are there any questions?
One question. Is that a leaf?
That would be a leaf, although it's going to be a little bit difficult because we need something to be flat. So remember, some leaves are highly specialized. So when you see a pine tree, those needles are actually leaves, right? They're just really specialized.
So when you make
And even though they're leaves, for this particular experiment, it may be a little bit difficult, so we need something nice and flat. So choose something flat that you can grab and take your sample from.
So when you
So I can see there are plenty, really nice large one right there. A lot of really good samples. I'm very happy that you brought your samples. So assistants, please come to the front. We're going to do it together. You're going to go back to your table, and you're going to assist your table the steps for this particular experiment.
So when you make your
As I set up my sample, I'm going to project it on this microscope, and there are a few light microscopes around the room that each table can use to see your own samples and data.
And when you
make your chart, you want to have at least 3 samples. You want to put the name of the species, and you want to draw the density of the stomata that you see and count them, literally count them to figure out the density. If you know the size of your sample, you can very easily calculate your density.
다 단다 교랑소 장도 지위 가서 름바든데 치료체스 되게 나로라 세신기 민다 되게 니리 카리 이미 디티야체 되게 나로 로마기다 용탈라야 다다 천시 솜라죠 로마 당보니 바 솜바체 되게 나로라 교랑소 뚝세 세야르다 교랑 천시 딘 나로라 되니 가제 이미 하고서나 젤라 뚝세 세야 딜렐하 도레스
And you can figure out different samples for each leaf to basically increase your accuracy. You basically have different densities, calculate them, and that will be your sample for that particular species.
다 단다 니리 찌긴바 이네 양스 교랑 니리 찌기 나로라야 름바 당보니 바 솜바 도스 요바 이나스 딜라 사말라 데스 체바 도라 데레야 촌돔 동알라 데네 교랑소기 tum데 딜라야 딱다 테야 저라 책토보 테야라 도스 펜도 요레스
Excellent. So we'll get started. Assistants, if you want to come to the front. In the meantime, start organizing your samples. It should take a few minutes, then go back to the tables and do your steps for your species. Okay?
다 단다 교랑소기 디 who are the teachers?
Assistants for each table. They want to come and watch me do it.
다 단다 단다 초라 로카레레 나로 단다 게겐 레로 쟁이 저라 도서 야 레로 제스 텔라 데네 데니 마 페체 초라소 쳐다 지아레스
We're good?
So 차소스
All right, let's get started. I'm going to borrow a few samples from this bag, and then who took this bag? Who?
디나로 단다 천시 카레 레야 디 수가 지레스 디디 단다 게겐 라고 주도 디 수레
Yeah. So when I finish, you can take it back. I'm going to take a few samples just to do it and watch the assistants watch me do it. Okay?
디나로 천시 카레 랭긴스 디니 교랑소 파피고스
Perfect. All right. Oh, that'd be great. Yes. Thank you. All right. Out for a second. Okay, things that we will use are polish, tape, and slides for other slides. Oh, I didn't see that. Right here. Okay. So this is for the other experiment. So tape, polish, slides, and like that's it. So we're going to take a few samples, and you want something nice and flat. So you think it's a and you will pour a couple. So you always want to do the back side, that's the front, this is the back. So do it on the back side. So get your samples, choose them. You want to be nice and big. You want to do at least 2 centimeters by 2 centimeters. A nice big square, about the size of the body. Let's do another, would be, we'll do another. First you want to do this. Nice and thick. You let it dry. Give it down a few minutes. Try not to do the veins. You can, but the veins usually don't have a good amount, so try to do it between the veins. just let it sit for a few minutes. Very gently. Let it dry. Try nail polish in a couple minutes. Yesterday I did it. It took about say 3 to 4 minutes. Try to do it. Oh, because we are going to take an imprint of the backside of the leaf with the tape. Once it dries, you take the tape and you do it around. Okay? And you put the tape on and you put it on the back. And you read the imprint. Okay? And you're essentially using the nail polish to allow you to take an imprint of the backside. The more that it dries, the better. So let it sit, let it dry. Just start making ink, right?
Is there any effect of this?
Is there one?
This, you apply this one. This doesn't affect
No, you just take it, get an imprint. You want to make sure that the leaf is not damaged, that the leaf is in good shape, it's not broken or bent, it's alive. No. And then what you want to do is you want to take a couple of good pieces. Scissors. Thank you. Keep it close to dry right now. So just take a couple of good pieces. And just put them on the table like this. Got it nice and ready. But you experiment. This is the way that we measure, the imprint. So it should be dry. Grab the first tape. And tape the area with the nail polish. Okay? Just like this. Okay? And gently push it in. Gently push it in. Don't do it too hard.
Smash.
Yeah, don't smash. You do the same for the other.
Why we do at the back side, not the front?
Because that's where the stomata are. All the gas exchange happens on the back side of the leaf. On the front, you have the oils that protect the leaf from desiccation, not on the back side.
The front is more.
We'll get another piece of tape. This one. You could try the front, but you will not see anything.
How do we count the tomato?
I'll show you in 1 second. Okay. Just here. Make sure that you cover the area with, and you can see where you have the nail polish, right?
Yeah, usually. Yeah.
Very gently put it in. Okay. And now we're ready for our imprint. Can I have 2 more slides, please? We have one right here. Make sure that it's clean, without any oils or anything. Thank you. Very clean. Okay. And now what you do is very gently peel this out. And now you have the imprint.
I see.
You see? And you put the imprint on the slide. As flat as possible. And now you have the imprint. You see? Thank you. And you do the second one. Very nice imprint. You see? Beautiful imprint. And you put this on the slide. Okay. Last one, thank you. Very gently, you peel this out. You have the imprint. Put this on the slide. And now we're ready to go to the microscope. Where's the first one?
This one is first.
The first one is already here? No, that's not it. Where's my first? Oh, right here. Yeah, this one. So now we're ready to go to the microscope.
Oh, start.
And now you could actually go to the 10x. Yeah, let's do 10x. So now you could see, very easily see, all the stomata that we are looking. There is one right here, one right here, one right here. And you start counting, right? You basically start counting. So what you do is you choose different fields, right? So this is one, and then you randomly can go to another one,
I see.
right? Another one without too many folds or too many kinks. That one's kind of dirty. We'll choose another good one. Something maybe, that's a good one right there. That's good, right? And then you count. You find the focus and you count the number of stomata.
Each of this one is one stomata.
1, 2, exactly. And you could even see that one's open, that one's open, that one's open, that one's open, that one's open. A lot of gas exchanges happen.
I see. Open.
Any questions?
Can we try this one?
Yeah, of course. And what I'll do is I'll leave this up for students that they can come in, and I can show them what it looks like.
The first one is the big one, is this one.
Yeah.
Now the second one is. How many were tumors are different? Amazing.
Very dense.
Wow. 넘버 장마 야시 생으로 지알았어. 단다디. 장마 야시 생으로 지았어. 단다 교랑 속에 디 롱콩디 장가지 가서스로 디 장마 장가지고 말했어. 니니 마지 장가지로 디 장가디 톰저디당 니니 파 장가지지 내바도 장가지사로 니니 마당 파니데 장가 주샤와이나 되게 찬용되라 그랬어. 니 끼마도서 맞진내래 파 주샤네 디그렐스. 세워차상.
Okay. So, assistants, if you can take a seat, that way we can talk about the second experiment.
genso di shuru chestne tanto chadaniba drugins chadaniba.
So, first of all, did you enjoy the first experiment?
Tanda chada dambodalo chona chongsoyas chona.
Did everybody watch their stomata? Were you able to find stomata in your leaves?
Tanda kyonasu di loma nalo lungkung di rasoyas lungkura.
Excellent. For the giant leaf, can you just raise it so we can see it?
저서
The
loma
one in the
chambo
middle
shida
table?
di namle gyaro chedas. Loma chambo de. 어,
Did you find stomata from that leaf?
디 나로 룽고 라소에스. No, no.
I would be curious to see what they'd look like for such a gigantic leaf.
야나 디 나로 단다 룽고 예메고레 셴데 셰라도 디 로마 하잔 쳔보 도아스.
All right, so for the second experiment, as we see in the picture here, we're going to visualize the process of photosynthesis. Now, stomata in your leaves essentially are the organelles that allow the gas exchange, what goes in and what goes out. CO2 goes in, O2 goes out. Water goes in, and oxygen is released, as we saw from photosynthesis.
다 단다 디딱 제고 니바디 단다 디나 따와데 직과레 디 nga랑수기 두수수 삼노로 두스탕로 체스 페나 디나 차샤와이나 ngaso 외조데 칸데 조고에 메디나레스 디나로 단진 nga소기 로마나로 단다 룽코기 디 가르체고레 세와나 nga소기 게스트서 야마 동네 체여라 디기 샤와데 디 단다 디나 지거레스 외조데나.
Now, for this experiment, we're going to see the process of photosynthesis. So you'll be taking little punch holes of your leaves using straws, and I'll guide the assistants on how to do it.
다 단다 디나로 가르세와이나 ngaso 외조디 디나리야 외조기 림바 도단시라 디티야레스 디 가르시야레 세와이나 그랑서 단다 딘디직 나로라 로마 이군 친주 세로와 디 파게 나루니 제단치레스 디 단다 겐기 그랑서 이런 바땡 고스.
You're going to get a cup with water and baking soda. Why do we need baking soda?
다 단다 디나 가르세와이나 ngaso기 디 셰포직체 디기 나로라 ngaso 추체 디 나로 ngaso 베킹 소다요 셰메 딘디 루고레스 디네 디 베킹 소다 고야 디 좀세 가르 인사레스 디나 루고야 돼.
Anybody know why we have to add baking soda?
디나로 베킹 소다 루고야 저라 다지 디나로 좀세 가르 이메 하고 고요 베스. 라 야보 코마스 carbon dioxide to expose the carbon dioxide.
So what are the requirements for photosynthesis? Water and what else?
단단 가서 단단로 외조 정화라스 가르가르 고레스 추체 셔바 가르 고레스 carbon dioxide.
Where do we get carbon dioxide?
carbon
From
dioxide
baking soda.
carbon dioxide 가 라그레스 와이나 베킹 소다네 라그레스.
So if you don't add the baking soda, my friends,
That's sense.
if we don't add the baking soda, we would never see the rise of the leaves, right? So as they photosynthesize, they're going to exchange gas and they're going to start floating up. And that's the whole key to this experiment. We take our cup with baking soda, we make our leaves sink. I'll show the assistants how to do it, and then you will expose them to light. Use your phones to put the light through the cup. In a couple of minutes, you should see a couple of those discs start to rise up as they photosynthesize.
다 단다 딱지기 디나로 가르레스 와이나 단다 포바 디나로 출루라 디나로 외조 도단 데랑야서 예벌도 가르 고레스 와이나스 단다 carbon dioxide 단다 디마르 carbon dioxide 베킹 소다 루고레스 텔로와라 테네 가서 가르 통고레스 와네 디 강로 현황소고 외지 우스탄 와이나 디니야 단다 디야 도단 저라 디야 비교여 바데 가서 통도고레스.
So as they photosynthesize, they're exchanging gas. They're taking carbon dioxide in, and they're releasing oxygen. The density of the gases change, and they start to rise. Now, this video is sped up. It would not be at this speed. It will be much slower. Within 5 to 10 minutes, you should see some of the discs start to rise.
다 단단 가서기 외적이 디나로 가르레스 와나 게스트 야마 젤렌 체고레스 젤렌 시단 가르레스 와이나 담보 디나로 가서 carbon dioxide 야레 디니 마 가르려 연거레스 와이나 단도 초롱려 연거라스 주레기 디나로라 디니 디 투세 만다 위왕게 렘베 야레기 레스 단다 디나로 저거 쇼도스 단다 크란소 응스 통이거스 저거 덴데 통고 마레스로 갈레체니 야 도단 통고레스 까마 주스 나로라.
So at this point in your tables, start cleaning your desks. Get rid of the slides, start figuring out which leaves you're going to use for your experiment. I'll show the assistants how to perform this experiment. They'll go back to the tables and we'll do the experiment together.
다 단다 가르치야레스 와이나 수수로 칸나로 디 쪽세데 상마스로 체스 쪽세 디나로 디니 가르치야레스는 로마 마보 도아스 단다 다지 디나로 로마게 이군 춘주 비고야지 디 가르치고면 수수 로마데 담스 디 사와라 테네 수수기 기게 도소기 델레체 디 체단데 랍 디니 파도 단제나시 무티네 체야레스.
Good. So assistants, come with me. Everybody start cleaning your tables, organize your leaves for the next experiment.
다 단다 기긴서 대 페로 체스 되니 하마나 소소 쪽세 장마 소로 체스
What do you want to see? You want to see this? Which experiment did you like more, the first one or the second one? You like this one better?
Second one didn't try.
Hello, hello.
All right, my friends. So for the next few minutes, let's grab our seats and let's discuss the experiments that we did together.
那等到刚刚开始节了，我说大家起来，结果了这些经验。
So I just want to get a good idea. Which of both experiments did you enjoy the most? The first one or the second one?
那等到这话的干一生呢，但是猜到我说大家你钱一把，大家当铺的等到你把罗斯，第二年呢肯定装那钱我讲说是。The first one 哪个当铺英语哪个讲的 ？First one。
So raise your hand if you think the first one.
当铺的了装那毛英语哪个讲是？多少 one number one？
Raise your hand if you think the second one.
你把的装那钱我英语哦。
I think the second one wins.
啊，你把的 top 生是。
So more importantly, and I'm very happy to see that every single group had discs that were floating.
但等到我别个我讲说，等到如看恰恰那了这是把要讲了要定下是。
So just to recap and make sure that we understand, let's review what we did. We have water with baking soda. Why do we need baking soda?
다 단단 가서 접시도 채와이나 단체들에 네 포바지 나로 출루 되나로 베킹 소다 루로스 베킹 소다 루고야 점세 가르레스 수이네 carbon dioxide they have a carbon dioxide in there we need carbon dioxide
Right. You need that carbon for the carbon dioxide as a prerequisite for photosynthesis.
다가라레스 carbon dioxide 고레스 텔라 때네 가서 외조디 퉁와라 carbon dioxide 고레스
So you could, in theory, make an experiment where you have different versions with different densities of carbon dioxide, of baking soda. You may have different ratios of baking soda to test how that can influence the rate of photosynthesis.
다 단다 될랑 가서 딱지 가르쳐야 세바이나 단다 베킹 소다 마뉴라 빠베스라 투세 마뉴라 빠베 가서 외조디기 투야 슈체중 가제 로고 에메자라 될랑 가서 쳐다 지고 레스테 마뉴라 되데 될라 외조디 야 마뉴저라 슈체중라 쳐바 로고 레스
And if we think about the protocol of the experiment, why do they have to sync when we start the experiment?
다 단다 결혼서 저와지 간이 세바이나 단다 되나로 로마도서 가서 월마 딩고레 라소스 담보 딩지고다 딩고야 되 냥가데 가르레스
Anybody?
수이네스 랭교노저 to extract the air
Right. So when you have them on the plunger, right, you make the vacuum, you are sucking all the gas out, and they sink, right? So at the very beginning, they're going to be at the very bottom. They have no gas, and as they start to photosynthesize, they do the gas exchange. They start to release oxygen as they take all the carbon dioxide, and they become less dense, and they start to float as they photosynthesize. And that is exactly what we measure.
다 단다 다라레스 가서 디가슬이야 단지디 올 팅고야디 준세 간이 세와이나 단지 디나로 가서 룬도스 땡고야스 단지 가서 컵디나 룬야 땡고도스 디 데올라 때네 데니 디마 올 싱고레스 데 싱고라 때네 디 야레당데 간이 세와니 데나 카본 다이오사이드 디기 투세데 야야야 파올라 때네 야디 가르중오 주르스나 솔롱용오 렘베지네 주르고레스 디기 외조데레스
So you could, in theory, make an experiment with different cups of different species to test the rate of photosynthesis.
You mean the different cup and the
Different cups with different leaves from different plants. Yeah.
다 단단 가서 여라 단다 포바데양 만다와 대당 로마데양 만다와 데라 때네 가서 게 디툼데당 최샤데 여라 데 만다도 추고레스 툼데서라 제발
And why, another question for all of you, why do we have to shine the light from our phones? Why do we need light?
다 좋아지어다스 단다 디체위 가슬라스 가서기 카바기 로드 마땅거도스 디 왜디 고야 좀세 가르레스 We need energy to receive.
Absolutely. We need those photons. As we understood photosynthesis, those photons are coming in. They're exciting those pigment cells, those chloroplasts, right? And they send those electrons through photosystem 2, photosystem 1, Calvin cycle. We create glucose, and that is what we call photosynthesis.
다 단다 다가라레다스 교란 속에 단다 니메나 뉴발리를 라서 다가라레스 간이세와이나 외절레와라 토마 간이세와나 니메나로라 외도지 땅 외도디 단당 가서 로마나라 장제라 포 장제라 데라 데니 로도야 림벨레와라 데네 데니 가서 림바 망보치야 단다디 단다 림바 가서 타방데니 림바 니바다 당보치 다나시 캘빈기 코준 라이조라 림바 데라 데네 가서 가르 정거레 세와이나 단다디 외조기 림바 데레고레스
And let's see how well you remember from our first session. Without looking at your notes, in photosystem 2, what molecule do we create?
다 단다 혈안 속에 디델라 마다르 체스 단다디 the number 2
Photosystem 2.
단다디 외조기 림바 니바 지어라스 외조기 림바 니바 데나로 가서 그 뒤두지 뚱고 요아스 니바나 뒤두 트야 뒤두데 가르레스 NDPH That's it.
I'm hearing two answers.
레니 고도스
Raise your hand.
차장로 제스 됐다.
Raise your hand if you think NADPH.
혈안 속에 NDPH 레상계대 차장로 제스.
Okay?
NDPH.
Raise your hand if you think it's ATP.
ATP 레상계대 라고 장로 제스.
The right answer, drum roll, is, drum roll, ATP. And what molecule do we create in photosystem 1?
단다 데스로 단다 외적이 림바 당부 낭로라스 뒤들 카르팅그레스
NADPH.
NADPH
ATP and NADPH are used in what? The carbon cycle to create what?
다 단다 디 뒤두니 도아스 디 단다 까봉기 고린데 낭로 카르팅야 체더레스 카르소야 체더레 글루코스
Glucose. All right. Are there any questions about either experiments, experiment 1 or experiment 2?
다 단다 가서 최다 림바니 제소 최다 당보다 니바 단다 림바 지에 디니니단 제게 동디데 예베스 So do they have any, the measurement of the baking soda when you are put into that one?
The way that I did it, since we don't have a scale, I just had to do it by eye. To make it more accurate, you would measure an exact amount of grams to have a consistent amount of carbon dioxide.
다 단다 개인기 제와이 나 원내기 체제야 데리스 카메로 다 최최스 미 단위 롭스스 제네 두슬로바인스 준네 제와이 나 소소기 그랜 체게네로 상말로 직주 영향의 체도 예바도 체게네 두슬로고 와레스.
So you would measure the amount of water that you're putting in the cup and the amount of baking soda. So you would do about 100 mils of water, 1 gram of baking soda to have a consistent ratio. And you could change it to see whether the ratio has an impact on photosynthesis.
지단 단다 다지 제외 가서 단다 추인바 이내에 단다 백인 소다이나 최신 예진지 여라 상말로 최신 예지 직주도 수로고 와레스. 이내야 교란 속에 양 최신 만다 요왕게 양 좀리 휴지 태당 딘데 다지 제바이나 양 야빠야당 디기 니가 여라 만다와라 데네 디기 양 휴지 만다와 딘데 레야 소다 제야 여레스.
Very good question. Anything else? Anyone else?
So they have because of the thickness of the leaf,
Yes.
also that impact.
Absolutely, yes. The thickness may hold more gas, right? So it takes longer to suck the gas out. It may even take longer to photosynthesize and get the gas exchange. So you could do a hypothesis where you are testing the different thickness of leaf tissue to see the ratio of photosynthesis versus the thickness of the tissue. Very good point.
다가라레스 단다요 안속에 로마 툴리 왕기 단다 딜라 켜바 수고레스 페차샤와나 로마 투와 딘다로스로 단다 게스 마오라 딘데 송장 디 게스 땡 값을라 디 땡야디 깔레 카와당 디 마오레스 딘다 송장 딘디 투와 요바 송장 디기 단다 외조 레와라 디스 마옹고야 저라 딘데 요레스 양 교란속이 단다 단다 디 로마 툴리기 왕기 단다 외조디 톨레 칸디 조고메 딜라 야 다지 쳔다 디고레스
Gaelic, you have a
단다 디 코로나 베게 글루코스 소다 디 있잖아요. 코디니 추노샤 디 가서 베게 보달루 아니 왜 당신잖아. 니마 같이 받을 디 로마도 막감네 되기래서 그 문투네 마시네.
알레 두 단다 다지도 쳔
코로나 칼라드라데 와라다. 글루코스라고 와라다. 같이 받을 그 마시네 되기래서.
So this experiment we have put this leaf into this cup. So since we are just taking just giving light and everything,
Right.
So it seems like how long it can last it. So when it alive, so when it will, so since it get the sunlight energy there, so
Right.
he is how long it will last it.
until the leaf dies,
Oh,
a couple
yes.
of days, maybe a couple of days, depending on the species. Maybe a couple of hours. Eventually, the leaf, since it's not attached to a plant, it would die. It would stop working.
단다 디요 로마 디 단니마 카시나를 시도 그래 딜라양 로마 딜라양 로마 니리라 라레그레스 야 단다 디 코랑 지신다 데 카타서와 인장 그 베트단 네투고 마레스 니마 카시나도 시도 그레스
And you can also test the intensity of the light. Everybody used their phones. Somebody built a tower of phones. That was quite amusing, right? So the more light, the more radiation light you give, the faster the photosynthesis will be. And that is another trait, another variable that you can test in your experiment.
다 단다 딱지의 지 가서 교란소로가 카시기 카바직 베제치 외직 당해야 양 카시기 카바 망보 베제 당해 줘라 외 체중기 왕기 양 단다 외조 되게 야 슈체중 될라 양 제바 조거레스 양디양 가서 주시직 차고라 다제 제야 줘라 외 망뇽기 왕기 외조 될라 슈진 망뇽 조다 줘라 디양 단다 디 외기 체중 대양 단다 주시직 차고라 디 다제 제나 조거레스
You could, for example, test the difference between sunlight, putting it in the window, versus artificial light on a phone.
양 교란소 주시직 딱지 가르치야를 세워야나 실로게 랑중기 니미기 왜 대당스 양 아랑소기 미수기 단달로 덴데다와 당체 덴니니 빠루라 외절레야라 차바 가르 주고도 덴데 차다 첸데 지거는 주시 만다와 니기 차바 드른 지거레스.
And I have to say, I'm extremely happy and proud of all of you because this experiment is not easy and you all made it work.
다 드린 나라 가서 가오다 뽑아 쉬어스 간이 세워야나 시당 드린 당지 지야디 레라오 덴데 요 마레스 이네 교란스 차장에게 빼하고 정샤스 견라 빼 가서 정송스.
Thank you.
谢谢老师。
So, my friends, we are coming to an end for today. It's pretty much dinner time, so let's clean our stations, let's clean everything up, and enjoy the rest of the day, and we will see you tomorrow.
One question.
Oh, you have a question? Yes.
Tanda di nganza di loma di piki tanda chishe waltaya di tanda kachi yare ka tsum yare tanda patan kachi we sonda shitoa tagi geesh nyeme gusina nyeme gola ka tsum gire gondali chigirenda chisungsa.
So his question is we test into this microscope.
Yeah.
So this stematory is open or closed. Since you mentioned that in the daytime it is closed, seems like some of the open, but what is?
You will have a mix. You will have a mix. They're mostly closed during the daylight to retain the water. If you're open during the daylight, since it's hot, you're going to lose water. So most stomata are going to be closed.
다 단다 디나 차동 체세나 다야기 롱콩 러이다디 디 만체와디 니메 관라야 코 카게세 그레스로 이네야 창마 마레스 디 카게과에 가인스나 니마 사오슈 여바이나 고랑기 누바딘디 라도고라 된다 손자 만체와디 카게 그레스 이네야 창마 예베 예바요 마레스 제바요.
So usually at nighttime, they all open up and they do their gas exchange. They don't really lose a lot of water at night. It's cool, right?
단다 텐을 차샤와나 로마데나 단다 룽고데 카체그레스 델라데네 코란소기 디기 뉴발라도 가야 말에 대손장 코란기 게스트 소파즈 젤렌치야라 만차와 텐을 상마 카체그레스.
Excellent. Thank you, my friends.
데네 투샤나 단지 경이 될라 디 장마서로 제스라 데네 산이 제양스 라서